Microbiology, and I'll be focusing mainly on viruses, especially because of the relevance of viruses today. And I'll also be going a little bit over protists and bacteria and archaea. And all right, let me start now. All right, so first of all, it's helpful to start by defining what life is, because we're going to be talking about viruses, which are typically considered non-living. So it's helpful to draw like a barrier between life and things that aren't considered living. So biologists typically define living things as being able to uptake energy and use it for growth and development, being capable of reproduction, tending toward homeostasis, and being capable of evolution. And NASA has a slightly different and less specific definition of life, which is that life is a self-sustaining chemical system capable of Darwinian evolution. And this broader definition is reflected by the fact that life detection outside of Earth must be able to account for different characteristics than Terran life. All right, now moving on to viruses. So a virus is an infectious particle that's made up of genetic information stored inside a protein coat called a capsid. And viruses are not considered living by biologists and instead fall into a gray area between living and non-living. And this is because they do not possess the ability to replicate and perform metabolic processes without a host cell. And viruses are also not living by the NASA definition because they're not capable of self-sustained Darwinian evolution, and instead they evolve along with their hosts. All right, now I'll be talking about viral structure. So viruses can range in large numbers of sizes from anywhere from 20 to 1500 nanometers or 1.5 microns. And a virus basically consists of information in the form of nucleic acids surrounded by a capsid, which I talked about. And in some viruses, a lipid membrane can envelop the capsid. Viruses can store genetic information as double-stranded or single-stranded DNA or RNA. And a viral genome typically consists of a single circular nucleic acid molecule. And viruses and the viral capsids can be one of many shapes, including orb-shaped or polyhedral. Capsids are made from protein subunits called capsomeres. And some viruses have viral envelopes which are derived from the membrane of the host cell, so they contain the host cell lipids and membrane proteins. Viral envelopes also contain proteins and glycoproteins that originate from the virus themselves in addition to the host cell membrane proteins. And some of the more complex capsids are found among bacteriophage viruses or phages, which are viruses that infect bacteria. So on the right here, you can see the diversity of viral structure, including the tobacco mosaic virus, which has a helical capsid surrounding a single RNA molecule, adenoviruses, which have the icosahedral capsid with protein spikes at each vertex, influenza viruses, which are most similar to coronaviruses, which have the characteristic shape of a lipid membrane surrounded by glycoprotein spikes and RNA on the inside. And the last virus you can see is a T4 bacteriophage, which contains a really complex capsid with a head, a tail fibers, and a tail sheath. All right, so now I'll be talking about viral replication. So each kind of virus can infect cells of only a limited number of host species, which is called the host range of the virus. And the host range results from evolution of viral recognition systems. And these recognition systems are specifically the recognition of host cell receptor molecules on the membranes of the host cell by viral surface proteins, which are the spike proteins that you may have heard about. A virus can enter a host cell through different methods, including the injection of its nucleic acid molecules, endocytosis by the cell, or fusion of the viral plasma membrane with the cell's plasma membrane. And once inside the cell, the virus uses the cell's nucleotides, amino acids, ATP, and other molecules to make viral proteins and nucleic acids. DNA viruses used host cell DNA polymerase to replicate nucleic acids. On the other hand, RNA viruses use their own RNA polymerases. After production of the capsid proteins and viral nucleic acids, the virus self-assembles on its own. The lytic and lysogenic cycles are two methods of viral replication in phages and other double-stranded DNA viruses that are well studied. So in the lytic cycle, a virus infects a cell, replicates, and then lyses or bursts out of the cell, which causes the cell to die. So this just results in immediate death of the cell. And then the viruses that burst out of the cell can go on to infect more cells. And a phage that replicates using only the lytic cycle is known as a virulent phage. On the other hand, in the lysogenic cycle, a phage exists in a state of lysogeny, which means that it remains inside the host cell without killing the host cell for long periods of time. So a temperate phage is a phage that can alternate between the lytic and lysogenic cycles, and most cells that go through the lysogenic cycle are temperate phages because they can go back to the lytic state. Uh, so in the lysogenic cycle, what happens is that a phage inserts its DNA into the cell, and then the DNA forms a circular molecule. The phage DNA is then incorporated into the host cell's chromosome by using viral proteins that break both of the chromosomes and then joining them together. A viral DNA molecule incorporated into the host chromosome is known as a prophage. And one prophage gene prevents the others from being coded into viral proteins immediately by coding for a protein that prevents transcription of the genes. And this way, the prophage can remain dormant inside the host cell without replicating immediately, without being turned into the viruses immediately. So every time the host cell replicates, it also replicates the prophage so that the prophage is incorporated into both of the daughter cells that result from replication. And in this manner of like silent replication in which the prophage is just inside the host cell and does not cause any direct harm to it, lysogenic viruses can spread through many generations of cells. Uh, an external environmental signal, such as chemicals or radiation, can cause the virus to switch from the lysogenic to lytic cycle. And in addition to the prophage gene that prevents transcription of most other genes, a few other viral genes may be expressed by the cell even when it's still in the lysogenic cycle. And these expressed genes can alter the phenotype of the host by producing substances such as toxins or offering resistance to the antibiotics. And these toxins and resistance to the antibiotics can be used by the host cell to its own advantage. Right here on the right, you can see a diagram that shows both the lytic and lysogenic cycles, and you can see that in the lytic cycle, the virus just replicates inside the cell, then bursts out, killing the cell. Versus in the lysogenic cycle, it stays as a prophage and then replicates into many other cells, and then eventually it can transition back into the lytic cycle, and then burst and kill the cells. All right, now we'll be talking about prokaryotes. So prokaryotes are defined by their lack of a nucleus or or of membrane-bound organelles, and they mainly include organisms of the domains bacteria and archaea. Most prokaryotes are unicellular, but cells of certain prokaryotes can remain attached to each other after cell division. Uh, Most prokaryotes have cellular diameters of zero point 0.5 to 5 microns, and they come in a variety of shapes, but there are three main shapes, which are cosi or spear-shaped, bacilli or rod-shaped, and spiral-shaped. All right, prokaryote structure. So prokaryotic cells have cell walls, and these cell walls help maintain shape, provide protection, like physical protection, and also protect the cells from lysis and hypotonic environments in which lots of water will enter the cell. And these cell walls will help prevent the water inside the cell from causing it to burst. So cell walls of prokaryotes from the domain bacteria are composed of peptidoglycan, which is a polymer made up of sugars that are linked together by short polypeptides. And on the other hand, cell walls of prokaryotes from the domain archaea do not contain peptidoglycan and instead contain proteins and polysaccharides. Uh, Gram staining is a technique used to classify prokaryotes of the domain bacteria by their cell wall composition. In gram staining, bacteria are stained with crystal violet dye and then iodine, and then rinsed in an alcohol, and then they're stained with a red dye that enters the cell and binds to DNA. So the purpose of the crystal violet dye is to indicate one type of bacteria, and then the iodine causes the crystal violet dye to complex, and then which traps the crystal violet dye inside that bacteria. And then on the other hand, the red dye is used to identify another type of the bacteria which is not able to retain the crystal violet dye after washing in alcohol. So specifically, gram-positive bacteria have the cell wall made up of a single peptidoglycan layer, which retains the crystal violet dye, since it's a lot thicker than the peptidoglycan layer of gram-negative bacteria. On the other hand, gram-negative bacteria have a peptidoglycan wall that's surrounded by an outer membrane layer, and the outer membrane accounts for a lot of the thickness of the whole envelope, and thus the thin peptidoglycan cell wall of the gram-negative bacteria is unable to retain the crystal violet dye. So uh, instead of having a crystal violet color, it instead has the color of the other dye, which is the red dye that binds to the DNA. And so the gram staining is mainly used in the domain bacteria rather than archaea because most archaea stain gram negative in the gram test regardless of their cell wall composition. And this happens because their cell walls are so disrupted by the staining process that they're unable to retain the crystal violet dye. An exception to this is the archaea, Methanobacterium formicissum, whose cell walls contain pseudomurin, which is a chemical that closely resembles the composition of bacterial peptidoglycan. All right, moving on to protists. Protists are a diverse group of small unicellular eukaryotes. Uh, Protists used to be classified under the kingdom Protista, but this kingdom is now obsolete as taxonomists have come to realize that not all protists are closely related to each other. So instead of being used to refer to an entire kingdom, the term protist is now used by biologists as a convenient and general umbrella term to refer to unicellular eukaryotes that are not plants, animals, or fungi. So right here, you can see some of the examples of common protists, which include dinoflagellates, radiolarians, ciliates, euglenids, uh, diplomenids, centrohelids, and foraminiferans. Many of these protists are aquatic and are found in environments like the ocean or lakes and rivers. All right, so now I'll be talking about endosymbiosis by protists. So the diversity of the protists that I just talked about likely arose from endosymbiosis, which happens when one organism lives inside the cells of its host organism. Uh, so and this relationship of the one organism living inside the other is mutually beneficial to both of the cells. So structural, biochemical, and DNA-based data all provide evidence that mitochondria and plastids, which include chloroplasts, were of endosymbiotic origin and that they were engulfed by early eukaryotic cells. Evidence also suggests that mitochondria were present in eukaryotes before plastids. So studies that compared mitochondrial DNA from eukaryotes to DNA sequences in bacteria and archaea have found that mitochondria are most likely derived from an alpha proteobacterium. And this proteobacterium was most likely engulfed by an archaea, which then gave rise to eukaryotes once the archaea had mitochondria inside it. Uh, Plastids such as chloroplasts likely resulted from heterotrophic eukaryotes acquiring a cyanobacterium endosymbiote in addition to the mitochondria. And this gave rise to green algae and red algae, which have their own chloroplasts inside them. And secondary endosymbiosis occurs when a cell with an endosymbiont is engulfed by another cell. And it might be of interest to know that this endosymbiosis is also how our own mitochondria were derived from, because animals uh, evolved from a protist known as the coenoflagellate. All right, so right here on the right, you can see how endosymbiosis works. So in primary endosymbiosis, you have a heterotrophic eukaryote with a nucleus that engulfs the cyanobacterium, and then this and then depending on the type on what happens after that, this can form either red algae or green algae. And then in secondary endosymbiosis, you can see that the algae are engulfed by another organism, forming the double membrane of the chloroplast, uh, which is an important part of chloroplast structure. And you can see that after secondary endosymbiosis, that allowed for the organism to have a lot of diversity, and it eventually differentiates into a lot of the protists that we see today, such as the staminophiles and euglenids. All right, and lastly, I'll be talking about the ecological significance of protists. So most protists are aquatic and are thus found anywhere with water, and they can form symbiotic associations with other species. Uh, and some of these examples include the zooxanthellae, which are a type of dinoflagellate, and coral. And having the coral basically provide the zooxanthellae with a place to live. And in return, the zooxanthellae photosynthesize and can provide the coral with its color and uh, energy from photosynthesis. Uh, and then another example of a symbiotic relationship is between wood digesting protists such as trichonympha and termites. And these trichonympha allow the termites to digest cellulose, so the termites can eat wood and then obtain energy from it. Versus if they didn't have the trichonympha protists, they wouldn't be able to eat wood. So that's pretty beneficial to them, but obviously not good for us since it allows termites to eat our houses. And another example of symbiotic associations is between algae and fungi, which form lichen, which you can see pretty much everywhere around here. Symbiotic associations of protists and their hosts can also be parasitic and cause harm. And an example of this is Plasmodium and mosquitoes, in which the Plasmodium is a parasite. And the Plasmodium protist is also the organism that is directly responsible for malaria and for making mosquitoes one of the deadliest organisms in the world. Uh, Another example of a harmful relationship is between uh, Fiesteria shimwei, which is a dinoflagellate, and fish, which can cause harm to the fish. And also between Phytophthora ramorum, which is a stramenophile, which causes sudden oak death in oaks and other trees. And a relative of this stramenophile, which causes sudden oak death, which is Phytophthora infestans, causes potato late blight. And this is the potato blight that contributed to the Irish famine in the 19th century. And you can see how that's pretty important. So protists have definitely had a huge effect on humans, including other animals. Uh, Many protists are also important photosynthesizers and provide the base of the ecological food webs. And this is shown as about 30% of the world's photosynthesis is performed by diatoms, dinoflagellates, and multicellular algae and other aquatic protists. The growth of protists in aquatic environments is regulated by the availability of nutrients such as nitrogen, phosphorus, and iron. So human activities can greatly affect protist growth by altering nutrient availability. And this includes things such as the runoff of fertilizers, which can result in nitrogen and phosphorus being present in greater quantities than usual in the ocean, and that can have some negative consequences. And this is because increases in nutrient availability lead to the formation of anoxic dead zones, which can have a negative impact upon biodiversity. So dead zones basically occur when you have a lot of protists or algae or other types of algae in one area, and then you add lots of nutrients to that area. So the protists or algae begin to grow quite a bit because of the presence of a lot of nutrients. And then when there's a lot of those algae, you have you have heterotrophic bacteria which eat the algae, and then when they eat the algae, they consume a lot of the oxygen in the area, and they produce a lot of waste. So the combination of the depletion of oxygen and all the waste, all the organic matter that's present, causes more depletion of oxygen, which eventually results in a dead zone in which there's a minimal amount of oxygen, and this can be harmful to the biodiversity in that area. So you can see another example of how significant protists are to the environment, both negatively and positively. And global warming also affects protist growth by impairing nutrient upwelling, which reduces the growth of protists. And that's pretty much it for the lecture part of this meeting. And I have, once again, a very short quiz. So I'm going to stop sharing this and then share that. Wait, before that, I noticed that I have a quick question. So the question asked me was, are prophages how viruses evolve? And I believe, yes, this is true. Prophages, viruses can evolve from prophages can evolve through prophages because the virus being present in the cell as a prophage will allow for mutations to happen. I believe that they can also mutate while replicating on their own in the lytic cycle, because when they replicate on their own, that could also result in mutations, which could result in evolution. So yeah, replication of the DNA of the viral DNA as a prophage and as a whole virus can result in evolution in both ways. And yeah, thank you for the question. All right, here's the very short quiz. You can just join with this code at menti.com. All right, I tried to make these questions require a little more thought than just pure memorization questions. So I think it would be helpful if you took your time to answer them instead of just answering immediately like how you would in like Kahoots. All right, has everybody who wants to join joined? All right, I'll take that as a yes. So I guess I'll just start now. All right, yeah, excellent. Most people got this question correct. So, uh, the reason that this is the answer is because when you remove the cell's nucleic acid polymerase, the cell itself will first of all not be able to replicate. And since it's a DNA virus, the DNA viruses will most often require the cell's DNA polymerase to replicate itself. So removal of the DNA polymerase will result in both the DNA virus and the cell itself not being able to replicate. All right, yeah, so this one was a bit harder, so didn't expect most people to get it. Luckily, quite a few did, though, so that's good. So basically, for this question, you just remember that viruses will just infect organisms that are closely related to each other. So in this case, you just pick the organism that's most closely related to a sponge, and that is the coenoflagellate. So sponges, a part of sponges basically evolved from, which are the color cells, basically evolved from the coenoflagellates, and that's why it would be coenoflagellates. The other organisms are significantly further along the line in evolution than sponges. All right, yeah, this question was pretty difficult too, I think. So some people still got it right, which is nice. The reason, so right off the bat, you can eliminate mitochondria and chloroplasts. I believe I mentioned both of those when talking about endosymbiosis. Both of them were organisms that were engulfed. So the mitochondria was the uh, bacterium, and chloroplast was the cyanobacteria, and with the nucleus, you can versus the ribosome. So the nucleus has its own membrane, and therefore it's most likely to be another organism that's been engulfed versus the ribosome, which does not have its own membrane, and it's just composed of a bunch of protein subunits that are joined together rather than a whole organism. So from what I've read, I think that it's somewhat likely that the nucleus was an organism that was engulfed beforehand. We're not still 100% sure if this was true, but it's more likely to be an organism that was engulfed than the ribosome. And yeah, that's pretty much it. All right, congratulations to Yo Mama for winning. And that's pretty much it. Uh, if nobody has any other questions, we can end it here. So yeah, y'all can leave now. If anyone has any questions, feel free to stay and ask them privately, or if not, you can just go. Yes.